Welcome to Academia YouTube channel. In today's lecture, we will be discussing on Central of Molecular Biology. This topic will be divided into 3 parts and this video will be part 1. Thus we encourage you to subscribe to this channel, so that you won't miss the upcoming videos. Okay. Before we go any further into the topic, we need to understand what central dogma of molecular is. Central dogma of molecular biology is the process in which the genetic information flows from DNA to RNA, to make a functional product protein. The historical background of central dogma of molecular biology. This fundamental theory of central dogma was developed by Francis Crick in 1958. His version was a bit more global and included the notion that information does not flow from proteins to nucleic acids. Scientists have since discovered several exceptions to the theory. One particularly notable example is that of prions. Prions are infectious proteins which replicate without going through DNA or RNA intermediates. Prions are responsible for the rare but devastating neurologic disease, Creutzfeldt-Jakob, which is a uniformly lethal disease that causes degeneration of the nervous system. In molecular biology, central dogma illustrates the flow of genetic information from DNA to RNA to protein. It is defined as a process in which the information in DNA is converted into a functional product. It is suggested that the information present in a DNA is essential to make up all proteins and RNA acts as a messenger that carries information through the ribosomes. The central dogma illustrates the flow of genetic information in cells, the DNA replication, and coding for the RNA through the transcription process and further RNA codes for the proteins by translation. The new DNA strands are formed, with one strand of the parent DNA and the other is newly synthesized. This process is called semiconservative DNA replication. The central dogma takes place into 2 steps and these are 1. Transcription. 2. Translation. These steps will be discussed in depth in the next upcoming videos. Transcription. This can be defined as a process of copying DNA segment into RNA, or it's the process by which the information is transferred from one strand of the DNA to RNA by the enzyme RNA polymerase. The DNA strand which undergoes this process consists of 3 parts, namely promoter, structural gene, and a terminator. The DNA strand that synthesizes the RNA is called the template strand and the other strand is called the coding strand. The DNA-dependent RNA polymerase binds to the promoter and catalyzes the polymerization in the 3' to 5' direction. As it approaches the terminator sequence, it terminates and releases the newly synthesized RNA strand. The newly released RNA strand further undergoes post-transcriptional modifications. Translation. Translation, as related to genomics, is the process through which information encoded in messenger RNA (mRNA) directs the addition of amino acids during protein synthesis. Translation takes place on ribosomes in the cell cytoplasm, where mRNA is read and translated into the string of amino acid chains that make up the synthesized protein. It is an active process which requires energy. This energy is provided by the charged tRNA molecules. Ribosomes initiate the translation process. The ribosomes consist of a larger subunit and a smaller subunit. The larger subunit, in turn, consists of two tRNA molecules placed close enough so that peptide bond can be formed at the expense of enough energy. The mRNA enters the smaller subunit, which is then held by the tRNA molecules of the complementary codon present in the larger subunit. Thus, two codons are held by two tRNA molecules placed close to each other and a peptide bond is formed between them. As this process repeats, long polypeptide chains of amino acids are synthesized.